Okay, I'm so sorry I got disconnected with my previous discussion since my laptop got empty. So I'll just continue our discussion on this topic, vegetation analysis. So, okay, so we're done with this part. So, um, we also have no aside from stand and plotless kinds broad kinds of samples, no, we also have the quadrat, which is one of the very common samples or sampling that in biological analysis or sampling method. So for quadrat, this is the delineated area or sample unit in the ground, usually square or circle. So size depends again on the purpose and availability of funds. So basically in quadrat, you will only be making squares, having specific measurements, like for example, 1 meter by 1 meter quadrat, or maybe in 10 meter by 10 meter quadrat. Then you'll have sub quadrat inside the 10 meter by 10 meter quadrat. No, so it depends on you. And of course, size also depends again on the purpose and availability of funds. So if you're going to do quadrat sampling, then you also have to depend on your funds, because not all the time na kuan siya kanang makasustain ka sa imong funds. Although this kind of method is not very costly, but the area that you are going to sample is, of course, depends on your time and of your funds na sige kag balik-balik sa area tapos dili pa nimo siya mahuman. So for quadrat, usually when you use quadrat, it also specify the on herbaceous plants. So these plants are what we call from the word herbaceous lang siya, herb. So it does not have much wood and its stems are green and soft. So usually these are not woody plants, and these are also considered plants that grow fast and produce flowers and many seeds in a short period of time. So basically, it's not comparable with trees that are also called as annuals or biennials. So when we say annuals, they only grow, or they only, let's say, their growth is basically after a year. So annuals, biennials are every 2 years. Perennials are hundreds of years, for them to be alive, or to grow spontaneously. So again, for plotless samples, there is no definite shape. So basically, kung katong anak ko nga, kung mangita mo random lang na tree sa forest, so let's say, napilian na ninyo itong mga pinakadako nga tree. So all you have to determine is quadrat. So since ang quadrat is north, south, east, west, so basically you have 4 plots. So gikan sa imuhang gipili nga tree, sa imong random sampling, the 4 nearest plots should be measured, should be measured by in distance, from that particular plant or tree na imuhang gibutangan. So muna siya nga danha magsugod ang imuhang kining distance. So the sampling methods can either be random sampling, regular sampling and stratified random sampling. So basically, ato na siyang ipang-discuss karon. So what are this kind of sampling? Although you already had this in your statistics and by stat, so we'll have a quick review on it. So vegetation parameter is best determined by actual measurement. So it should have to be done. It should have to be, yes, done by actual measurement. So this is an example of random sampling. So for example, this is your area having 10 meter, 10 meter or 1 hectare land. So pwede ka magsampling dari, pwede pa ka magsampling dari, dara, bisan asa lang ana, yung kaning mga nay-X. So that is random sampling. So bisan asa, random. Okay, another is the regular sampling. So as you can see, regular sampling or what we call systematic sampling. So here, one of the number of samples are taken at regular intervals. So example, nagbutang ka dari og line, this is your first line, then definitely naa kay mga systematic siya. So dari na ni distance like 5, let's say 5 meters apart. So definitely mong 5 meters here, second 5 meter here, third 5 meter here, and so on and so forth. No, so maybe sa imuhang first 5 meter, you are in a grassland area. Then next 5 meters will be in the forest area. Then still the same, the forest area on the third 5 meter. Then the fourth 5 meter is the ipil-ipil plantation. So on and so forth in anang iyahang systematic, meaning sunod-sunod. So when you say systematic sampling, no, it is similar to simple random sampling, but it is usually slightly easier to conduct. Kaya since naamang tayo gisundan. Compare with random sampling, ikaw lang mahala pa ka sa kamagsugod, pwede ragay mo nga duol ra ka sa imong gisamplingan, kung saan po gani ka nagsampling, dan hira dahil tapad imong imong sunod. No? So definitely, it's your own choice when you say random sampling. So every member of the population is listed with a number, but instead of randomly generating numbers, individuals are chosen at regular intervals. So, dili ka mag-choose like 1 or kanibitang sunod na pod kay 5. So, sunod na pod kay 10. You can choose that 5, 10, no, or by 5s. Like 5, 10, 15, or maybe by 20s, 20, 40, 60, no, 80, so 100, so ana siya. So another is the stratified random sampling. So in stratified random sampling, as you can see, it is a type of sampling method in which the total population is divided into smaller groups or strata to complete the sampling process. So for example, when you say stratified random sampling, by strata siya. So meaning kung this is the whole hectare of your study site or study area, then definitely when you do stratified random sampling, makita ninyo nga sa grassland, nag-random ka pero sa grassland na. Sa forest, nag-random ka pero sa forest lang na siya. Ipil-ipil plantation, also, random sampling po ka pero within ipil-ipil plantation. So it's not like random sampling with, compared to random sampling, no, kaning random sampling is sa whole acre, sa whole hectare, no, gani ka nagsample sa forest, sunod na po sa grassland, sunod na po sa kaingin, sa secondary forest. So murag light siya compared to stratified random sampling. Stratified is every strata, let's say, kuan siya kaning for example in a population, let's say ato na siyang i-relate to population, like When you say stratified, when you sample population in a research study, no, you divide a sample, let's say adults, into subgroups. So subgroups by age, for example. So murag nabutang ka dito sa imuhang sample like from 18 to 29 years old, from 30 to 39 years old, from 40 to 49 years old, 50 to 59, 60 and above. Murag inana. When you say stratified, no, kanang naa siya layers. So naa siya groupings. So that's what we call stratified random sampling. So mura siya og kaning stratification, from the word stratification, it's the arrangement or classification of something into different groups. No? So lahi-lahi siya. Kung nag-random sampling ka, purely in ipil plantation, but not by doing random sampling in your overall hectare study area. So in anang stratified. So now let's move on to two broad types of vegetation parameters. So we have the absolute and non-absolute. So when you say absolute, so in this type of parameter, no, you'll be able to know the density, the cover, the biomass, and the performance of particular species in an area. But when you say non-absolute, frequency. Frequency is, of course, the number of occurrence of a repeating event per unit of time. When you say frequency, ikapila siya nagbalik-balik sa isa ka area or maybe sa isa ka kwadrat. Like for example, if you do quadrat sampling for 10 by 10 meter nga kwadrat, so definitely in 10 by 10, let's say kugon grass, ikapila ni balik si kugon grass within the 10 by 10 meter quadrat. So inana siya. Not absolute because ang frequency manggod dili na siya mag-alter. Kung for example si kugon grass dominated siya, let's say kuan siya murag 100 siya nga kabuok individuals for that particular species, then definitely dili na siya mabag-o. So lahi na pud tong number sa lain nga mga species. Like for example inani non-absolute kay mostly inani ang nakakita sa area, no? Inani tanan halusa. Unless kung move ka dito na lahi nga mga kuandari, plant species lahi po dari nga part, no? So inana. When you say absolute, no, you have to compute every detail of a particular species about its density, its cover, its biomass, and performance because they have differences manggod. Mag-alter ang iyahang kuan, ang iyahang result for absolute. Okay. Kung may tag non-absolute again, it's kuan, dependent on something else. So kibali, kung for example, the-dominate ang kugon grass, then definitely ang result nato will depend on the frequency or the kanang ikapila siya nag-occur in nga area. So, di na gud siya mabag-o. Okay, since dagan man ang kugon grass in nga area, for example. Okay, now let's have the definition of terms, no? When we say density, it is the number of species in a given area. So, it's by way of counting number of plants, divide by sample area or quadrat. So inana ang iyahang computation. But we'll have more on the computation on the later part of this discussion. So another definition of term, we have the cover. So when we say cover, literally, yes, the one that covers, dili lang siya na to is specific into cover sa taas, but also cover in terms sa ground. So it is the proportion of ground occupied by perpendicular projections of the aerial plans of individuals of the species under consideration. So it is normally expressed in a percent. So kining cover manggod is defined in every species in which kung ang kaning mga species are living on the ground, then definitely you also have to determine their cover. on the ground. But there are also cover nga, let's say, specific to trees. So usually in our, if I can remember, no, when I was in college, no, we do sampling cover, percent cover of canopy, forest canopy. So all we have to do, kasi since wala man kay may complete nga gamit na dala adto, so we use kanang tissue bitaw nga kana bitaw pagmahurot ng tissue kana iyahang murag cardboard bitaw, cardboard niya. Dayon butangan na mo siya thread and himuan na mo siya og kanang kwadrat, murag bitaw siya og thread nga murag mag-cross sa circle. Then all we have to do is to peep or to look look through the patong buslot sa tissue in order for us to estimate. That's what we call estimation of percent cover. So kung for example mo igo siya sa isa ka kwadrat fully, then definitely it's 25% cover, no, kana siya. So estimation lang ang tawag ana, but there is also computation for percent cover. You'll discover that later. Okay, so we have biomass or the yield. So it is measured by clipping the above ground parts of each plant species and oven drying them to constant weight. So you'll have the procedure later. Then we also have performance. Common measures are leaf length or leaf width, flower number, length of flowers, flowering spike, number of seeds per panicle, measures of reproductive performance. So panicle is the measures of reproductive performance. So basically, it's all about the health, no, of a particular species that it is defined or it measures the leaf length, width, and flower. It also describes the flower number, length of the flower spike to know whether kung kani siya nga particular plant species is kuan pa ba siya, immature pa ba siya nga plant, or juvenile pa ba siya nga plant, or maybe adult na siya. So another, no, again, is the frequency. It's the probability of finding a species within a quadrat when a quadrat is placed on the ground. So frequency, kapila siya nibalik sa isa quadrat, yes. So of course, frequency is affected by, first is quadrat size. So we cannot say that dapat sa isa ka quadrat dapat balik-balik. No, it's because we depend on the quadrat size. Let's say if you're aiming for 1 hectare nga isampling nimo, definitely lahi-lahi ang area nga makalap nimo tungod sa kadako. Aside from that, we also have to consider that not all areas, no, contain or has the same quality, same quality in terms of their soil. So definitely, dili pasabot na makita nimo siya dari, makita po nimo siya sa lain nga imong gibuhatan sa imong isa ka hectare. So it depends on the quality or the physical and chemical quality of the soil. So maybe kung dili siya makasurvive kana nga particular species ang katong pikas nga medyo dry kaayo og land or soil, then definitely dili siya present dito. So that's why frequency is affected, na dili siya makita. Makita siya dari sa pikas, pero dito sa pikas dili na siya makita. So that's it. So next is individual plant size. So yes, we also have to consider frequency in that case because not all plants will grow. Let's say, any mga particular plants can grow, let's say, 50 centimeters or maybe 50 to 100 centimeters. Then definitely not all plants have the same plant size. Maybe also have to consider the quality of the soil. So basin dili kayo siya gakatubigan dari nga part, then dito sa part kay duol siya sa kuan kaning mga river or sa basin ba kay river basin or something like that, then definitely they have present sila in a way dili sila parehas of sizes. So dili siya ma-consider siya nga present gyapon siya ana nga area pero dili pasabot nga i-alter nimo ang frequency. So definitely basta na ang presence niya, then you can always account that for frequency. So another is the spatial distribution of individual plants. Like I've always mentioned that plants can be dispersed all over the area. So it depends kung asa lang sila makathrive for that specific area because wala to kabalo basin dili suit sa ilaha kana siya nga habitat. So that's why nga dili kay sila makita dara pero naa sila sa pikas. So muna siya spatial distribution kung asa sila na distribute nga mga places in, for example, in 1 hectare. So maybe na sila sa upland nga 1 hectare, ang uban din naa sa lowland. So frequency is affected by that kaning distribution, spatial distribution. So here, the dependence of frequency on quadrat size. So kani siya, this is an example, frequency on quadrat size. So for example, this is the quadrat. Now as you can see, species found here in this part will not be maybe found here or here, but they are on the same quadrat. So mura na siya ang iyahang ibig sabihin dependence of frequency on quadrat size. So maybe kani siya nga species dari, makita nimo siya dari, ano nga part, pero dari nga part, di na nimo siya makita. Maybe, pwede na siya, possible. Another is the dependence of frequency on plant size. So maybe if this is your quadrat, 10 by 10 quadrat, then definitely nay gamay dari, nay dako dari, gagmay dari, no? Dagko. So yes, it doesn't matter. As long as there is the presence of this kind of species in that quadrat, then definitely you'll account that for as frequency. So another is the dependence of frequency on spatial pattern. So there are different types of patterns, no, spatial, space and time. Naiuban, sa isa ka quadrat, nagtapok sila. So, kana siya, si kugon grass na puni, kugon grass, sila tanan, kugon grass dara. But there, in other quadrats, maybe nagtapok sila. Clumping, that's what we call clumping. No, nata na kugon grass dara, for example. Another is, nagtapok sila but they are evenly distributed. So as you can see, the presence of kugon grass here can also be seen here, then here, and other parts of the quadrat. So always think na kada quadrat, lahi-lahi ang iyang distribution, spatial distribution. So that's what we call. Okay, now we'll move on to the vegetational analysis in grassland. So why do we do vegetational analysis? First is characterization of plant community at any one time. So when we characterize, we are not just only doing soil quality and all. We also characterize species that are present in the area. So, kay ano na siya? Because not all plants thrive in one soil, one condition of soil. So, kung mayroon ka gloomy soil, sandy soil, then definitely there are particular species that can only thrive in that particular soil type. So, dili tanan mabuhi. What na siya? Same goes with if, for example, makita, let's say strawberry or maybe potato can only be planted in above sea level, let's say 100 meter above sea level or 100 feet above sea level. I mean, yes, 100 feet. So definitely, dili mo makita or matanom dari ang strawberry farm ug ang potato because they need particular soil type as well as the condition of the weather or yes, the weather type sa ilahan, no. For example, sa mga bukid-bukid, usually mga strawberries are much kining can only thrive on cold areas. So definitely sa mga bukid-bukid sila present lang. So dili mo sila makita dari sa ang strawberry farm dari sa Cagayan na naagi dari, no. So usually sa bukid-bukid gyud na siya. So another is comparison with other plant communities. So in this way, by doing vegetational analysis, we can compare other plant communities in such ways that definitely in vegetational analysis, no, we do not only focus on one area, but we compare all areas nga kalap siya sa isa ka, let's say, isa ka hectare. I'm not saying that in 1 hectare equal tanan ang soil type. So maybe sa 1 hectare na kalap sa imuhang land area na ay lowland and upland, then definitely there are comparison nga dili tanan nga naa sa lowland present sa highlands, no, sa imong 1 hectare na area. So definitely na ay mga particular species lang nga mag-thrive sa upland o mag-thrive pod sa lowland. So dili sila equal og vegetation, no. So another is permanent record of its nature and occurrence. So definitely this is one of awareness to the locality that there are plant communities in their area na maybe rare, endemic, critically endangered or endangered, and then maybe kuan kanang or maybe least concern siya, sagbot lang siya. So murag nang para sa nang if we can try to look on Facebook nga ang mga kuan silang Gretchen Pulido na sila yung mga gabi-gabi sa ato kay katul kaayo na siya na mapunitra na siya bisan asa dito sa bukid. But since sa ila wala na kayo trees because all of their kuan areas there are converted into residential and other establishments, then definitely ignorante kay sila ana, no, murag wala na sila kabalo nga ay di ay sagbot taday na dito sa Mindanao. So of course na that's how nga that particular plant can only be found in, let's say, Mindanao only. So in ana then katong Rafflesia, for example, is endemic in Davao maybe, so in ana siya. Those are just kanang permanent record, no, that the DNR can use, no. So another is understanding of its patterns, functions, and dynamics. So in an area where this particular species are present, then definitely they have a relationship towards particular species in the area. So kung ignon nato nga daghan kaayo sunflower or yes, no, kanang mga kuan dara nga part. So definitely daghan mga bees na kuan or mga pollinators maybe na present in that area. So they have to this pollinators will disperse the seeds and then distribute it to other parts of the area. Then dito na po dahil manubot ng uban. So ana, no, kanang kuan lang that it's a relationship now. We have ecology now, the relationship of species to the environment. So another is the indicator of soil state, soil water status, and productivity. So definitely when you do vegetational analysis, you cannot do vegetational analysis when the area is degraded. Let's say na-convert na siya into kuan kaning land area lang tapos wala na siya laing vegetation kay you cannot do vegetation analysis magud kung wala kay makita nga vegetation didto nga mga plants. What is the purpose of your vegetation analysis if the area is being converted na into residential, for example, or agricultural land area? So usa ang pagtan-aw nimo sa vegetation. So, and of course, indicator of soil. You can only do vegetational analysis when you also test soil quality and other soil status and productivity in the area, because makapaas man ka nga, hala, anong daghan man kay dari nga kuan kani nga mga plants tapos gamay ra pud kay dari labon didto kay daghan kayo didto nya dari gamay lang. So definitely we can always compare the results of the soil quality part no nga lahi ang ilahang soil quality. So maybe it's an indicator in which kani nga area is very suitable for that particular species, or kani nga area is very kining harmful for the species. So by that way, no, makabalo ka. So kana siya nga unsay mga dili pwede tamnan dara kay kung itanom na nimo mura siya og kanang bitang invasive siya na nga area, so kung mudaghan sila then definitely mag-dominate na siya sa area and then causing it na mawala tong uban ng mga species nga iyang tapad. So that's it. And then we also have determination of community diversity. So by this now we can be able to know what are the different species that are available in that area. So we'll know also the conservation status, or the IUCN, the International Union of Conservation of Nature, in which kung kaning mga plants are endemic, rare, critically endangered, or maybe extinct. And then we also have identification of endangered and threatened species in the area. So IUCN Yapon. So for the sampling methods, now we move on to quadrat method, biomass or harvest. No, it's either in a way of how we obtain biomass or by way of harvesting, then ato siyang kuwaon na iyang biomass. So what's the size of quadrat? So there is, for quadrat method, no, there is what we call determining the species area curve. So I will be giving you a video. I will upload it in UStep, so that you'll know how to determine or to understand what species area curve is. So this species area curve is the usefulness of this concept lies on the premise that there is a minimal area which can be used for sampling. So this minimum area represents the characteristic species composition of the plant community. So kaning ang end point lang ang species area curve is you stop sampling when you reach to the point nga wala na kay makita nga laing bag-o nga species in that particular quadrat. Now, for example, sa quadrat 1, kita ka gugon grass, kita ka og ipil-ipil, kita ka og blah blah blah. Sunod na pud kay kita na pud ka gugon grass, ipil-ipil, and blah blah blah. Tapos dito na pud sa imuhang kuan, kita na pud ka gugon grass and ipil-ipil. So kung si gugon grass and ipil-ipil maura ang nakita for the whole quadrat, whole subquadrats, then definitely you have reached your species area curve. So, meaning you stop sampling because you reach plateau na, you stop sampling the quadrat areas, then muna din to. Si Kugon grass and ipil-ipil ra di ay nga makita dara sa imuhang whole overall na quadrat. So the procedure is first you need to have meter tape and then the string or rope. Then you set up quadrats in a manner illustrated by using the following dimensions. So for example, sa letter A, let's say, igno nato no nga si 4 by 4 meter, ay 8 by 8 meter ang overall na quadrat. So in your overall quadrat, you have to place subquadrats in a way nga inani ang measurement that can be divided into 8 by 8 meter. So, si 0.5 by 1 meter. As you can see, natay kuan na rin, no? 0.5, this one, by 1. Yes, this one. Another is 1 by 1 meter. You can see 1 here by 1 meter. Second is 1 by 2 meter. So, you can see here 1 by 2 meter. Another is 2 by 2, letter D, 2 by 2. Nisha. This is 2 by 2. And so on and so forth. So that's how you do subquadrats. So your main quadrat is 8 by 8 meter. So definitely you'll have to set up subquadrats for that. Para maibalaan mo sa mga kung sa imo ang pwede nga masampling dara o sa mga particular species ang present dara. So that's it. Now after that, you list species encountered in different quadrats. So starting with quadrat A, record only the additional species encountered using the table below. Number of individuals per species is regarded. So definitely when you do quadrat method, you only list down the species that are new. So kung na-encounter na nimo siya from the previous quadrats, then definitely do not write it down. Only record those species that are additional na imong na-encounter inside your quadrat. So this is a sample raw table for species area curve. So as you can see, you can write the species here. And then we have 2, 4, 6, 8 quadrats in which pila kabuok, no? Only ang imuhang ibutang dari is in your species area curve, you only have to indicate check or maybe asterisk. Check if it indicates presence, pero kung mo siya asterisk or diamond ba siya, it indicates absence. So, kung for example, Kugon grass is present in A, B, C, D, E, F, G, H, then definitely you check that. Presence versus absence, no, of particular species in an area, in a quadrat, I mean. So now, once you have already write down all the species you have found in every quadrat, no? Then you need to construct a species curve and determine numerical area by going through the following steps. Plot value of cumulative number of species against total area. Using 10% number of species and plots, plot A as reference line, then designate this as A1. Then draw a line parallel to A1 and tangent to the species area curve. The point where this tangent touch the curve is the lower range of the minimal area. So I know this is very confusing, you know, if you try to read this, medyo confusing, but I will give you a video, a full video. It's a 12-minute video maybe, I guess so, in which it only talks about species area curve. So para makabalo mo sa ano siya pagbuhat ang species area curve. So we will not be dealing with that here sa atuang discussion because we have 115 slides for this topic. It's very long, biological parameters, manggod, or methods. So we still have two more topics. So we haven't finished with vegetation analysis yet. So, okay. Next is example of data determining the minimal area of a pasture. Like for example, in subplot A, that is 0.5 meter or square meters or meter squared, no? Then these are the particular species present in the area. So, kana siya. Sa 1 square meter, then definitely kani siya ang mga present in the area. So, nakita ninyo nga kani si size 0.5. Ang nakita na dari kay si Arvense. Arvense. Si Arvense. Arvense. So, this cumulative total number of species is kuano kanimitaw dili na siya number sa pila ra ang nakita. At first, this is the basis for number nga pila ra ang nakita nga species dara. But when you try to see the video on species area curve, na siya formula in which nga katong nakita nga gabalik-balik ginamay-nusa na dayon siya. So you'll have that no when I upload it. Okay, example of data for determining the minimal area of pasture, no? Continuation for the previous slide. So as you can see, these are the subplots that we've mentioned a while ago on the 8 by 8 meter. So definitely, ano siya? May mga particular species that are present in the area that are not wala siya frequency because every time no when you move to another subplots there are species that are not the same with other species na nakita sa laing subplot. So definitely originally present halos ni sila but dili kay sila wala kay sila occurrence in the same subplots. Okay, here is an example of the graph of species area curve. I know this is a bit confusing, but as I give you the video on a species area curve, you will you will be enlightened by the video and kana siya no. It's really this kind of discussion is very in-depth. So I guess I have to suggest that you should watch the video. So kaning curve no, it's not necessarily it should be curved. You should follow the points or dots na gusto ninyo siya mapurma nga inana. But definitely all you have to do kay na may uban na nga mga points dari, mga dots dari na bulag na scatter, but you don't have to follow those scattered lines. Your goal is to form a curved line wherein no makita niya ang kaning tangent na line nga in which nga kung unsa ang maigo ani nga part, like this is the A2 and the B2, tangent parallel to the 10% and 5% line. So basically, kung maigo na siya, that is what we call your tangent line, this one, the broken lines. So for species area curve is the relationship between the area of the habitat and the number of species found within that area. So basically, kung asa sila present ang nga area, maupod mo, ihapon po nimo ang number sa species na makita ang nga area. So the relative numbers follow, then of course systematic mathematical relationships, then you'll be able to form a graph of species area curve from this out from this table, no? So you also need to follow the curved line, but not necessarily touching perfectly on the dots. Kay, as long as naka-form kag curve line. Then as you can see, once you reach to that point in which na the straight na siya sa curve line niya, na straight na siya, then that's what we call, you have already reached your species area curve. Na no new species are added to the area. So that's it. Now we move on to harvest method proper. So they use the right size of quadrat obtained from the species area curve. So kato ang size of quadrat. Then determine the number of quadrat as follows. So basically, you will not necessarily apply this coefficient variable. Pero kung gamay lang ang inyong i-sample, then definitely you'll be needing this. But when you do large sampling or when you do sampling in large areas, then definitely you'll no longer be dealing with this. Kay it's a very large scope in which na maglisod na mo o gamit og inani. Kay tinamban lamang gud ni siya. So all you have to do, kaning harvest method proper is only used for biomass. So, igo ra ka magkuha, i-harvest nimo, then all you have to do is, I just have to give a short background of biomass now. So, basically, biomass is a renewable energy source because the energy it contains comes from the sun. So basically, humans and plants has a percentage of biomass. So as long as we give off energy, then we have that presence of biomass. So the most common biomass materials used for energy are the plants. So sa mga researchers, they say that corn and soy are one of the biggest or one of the common biomass used for energy. They can, kaning mga energy from these organisms can be, can burn to create heat or are converted into electricity. So this can be, this can create heat directly, convert into electricity. It's a direct way and process into biofuel. So kung biofuel, maybe kani atong mga gasolina, pwede na nato ni siya ma-convert into biofuel in the future so that dili na kayo para environmental friendly na ang ato ang atmosphere. So by that, yes. So, of course, measuring biomass from field quadrats is difficult and time-consuming, especially when you have a bigger scope of land area. So definitely, you'll have to measure all the biomass available in the plants of that particular area na imong sample. So, aside from that, the basic technique of measuring biomass is the harvest method. So all you have to do is, of course, harvest all plants in each quadrat. So before that, distribute the quadrats at random in case of heterogeneous vegetation while stratifying the homogeneous area when getting your samples. So it harvests all the plants in each quadrat. You have to harvest all the plants in each quadrat. Then classify the harvested plants according to species. So kung pugon grass lang yung tanan ibutang dito. Then you have to dry it in an oven or under the sun. Then compute the importance value or dominance of the species using this formula. So dry weight of species A, like for example, pugon grass is species A, then divide by the dry weight of all species. So when you say all species, tanan, pugon grass, ipil-ipil, blah, blah, blah. So, pila ka count ana. Pila ka box species, let's say, nakita ninyo sa isa ka land area is 10 species, then you have to divide it by 10. And then, you multiply it to 100. So this biomass no is and basically it's removing or simply removing biomass by doing or by removing water and weight, no, by drying it, no. And then of course water or what yes water content of plants can vary dramatically day to day and even hour to hour, or so usually it is, I mean, the dry weight is more stable measure of biomass than in fresh weight. So kaning fresh weight, napa na siya mga tubig-tubig moisture inside it. So dili pa kay siya in anak ka stable. So aside from that, no, there are also choices in whether how you measure the total biomass. So it's what we call the above ground and below ground, or just above ground biomass. So most biomass studies in vegetation science, ilang gina-focus is on above ground biomass. So kani siya no, for the reason that ang kaning mga below ground biomass is very difficult to measure. So kailangan pa nimo siya ang sa term ana i-plow pa nimo ang soil para lang imong paon ang katong roots and all, then i-apply to nimo siya sa dry weight. So kaning biomass na above ground, those that can only be seen above ground can be measured, can be measured in biomass. So the stem itself, the leaf, and whatever nga nakita sa above ground. So wala na siyang measurement niya. So let's now move on to line intercept method. So it is useful in sampling non-porous vegetation types, especially if destructive sampling is to be avoided. And all standard vegetational measurements can be obtained in this technique, except absolute density. Now, how do we do line intercept method? So the procedure is you lay out transect line using a rope or chain. So for example, having this kind of, let's say, kani siya, muna siya yung transect line kana, forma na nimo dara. Tapos let's say 30 meters. 30 meters ang imuhang ang imuhang line, 30 to 50 meters. Then you subdivide this transect line into segments of any length to determine frequency. So maybe this 30 meters, now you have to divide it by, let's say, by 5. Let's say 5, 10, 15, and so on. So, Dani, sa imong first na transect, you have to determine what species can you find. So, ang ginagamit sa line intercept method is less destructive siya. So, dili ka magkalot-kalot, kailangan ng ginana. All you have to do is to make an observation. So, gikan na nga line, going to this line, you have to observe what can you see around you. Nga, igo raman ka nagbutang og line danha, then kung once you reach to this particular transect line, yung gibutangan nimong segment of any length, then you determine the frequency. Kapila siya ni balik. For example, the kugon grass, kapila siya ni balik dara. Kalima ba siya sa imong nakita within that line. Then followed by the second segment, kani na po ka magtanaw kung sa imong mga nakita. For example, tree dara, kugon grass ba dara, no? Ana. Or maybe kanang mga ano dara, kining mga leaf, something like that, and so on. Okay, so as you go along the transect line, record the species that underlie or overlie on intercept along the transect line. So this is an example. Oh no. Okay, this is an example of your recording data in line intercept method. So you have the species, the intercepted length. So, unsa to nga length ang imong gitumban? Let's say 5 meters out from 30-meter intercept. Then you have the total number of kining species found. Then you have the percent cover, which is not, kuan na siya, api lang siya dari, but later on will be answered. Kaya wala pa man ta dari nga part. Then height, you have to record your height and your average height. So for example, if you found, let's say 6 kabuok nga kugon grass, then iyahang height is between 8 and 7. So mostly katong, let's say, ang tulo ani sa 6 na total is 8, having 8 cm, and the other 3 is having 7 cm. Then definitely you have to know the average height. So, kana siya. So, where am I now? Okay. So on and so forth now. As a rule of thumb, if you can remember, the more diverse the vegetation, the longer the line should be. No, para makabalo ka kung asa pa ma-extend ang plant species, no. So kung daghan ka makita nga vegetation, then you have to extend your line intercept. It's not necessarily nga maura gid na imo i-set up na line like 30 meters lang, 50 meters lang. But if you can see that there is the presence of or more vegetation, then you have to increase the length of your line. Aside from that, no, the more homogeneous the area, the shorter and the less number of lines should be used. When we say homogeneous, kung gamay ra kayo mo makita sa isa ka area, pagamyan nimo ang imuhang line. For example, 30 meters lang gyapon na din sa unahan, pag after 20 meters, land naman di ay na. So you have to adjust your line into shorter or less number of line ng imuhang ibutang dara. Kay mura man gyapon wala gyapon siya pulos after 20 meters or so because wala na vegetation nga makita. So kana siya. So these are the following formula on determining relative height. We also have dominance or cover, relative dominance, frequency, relative frequency, then total coverage. So you'll be using this when you deal with plant species or vegetation analysis. So kani siya, you don't have to actually memorize this. You can always do manipulation in Excel. Tapos i-enter lang nimo ni siya nga formula, and then when you're done, you'll have to drag the cell, and then it will eventually give you the following answers to all species. So for example, in if individual species. So after that, computing those formulas, you also have to compare the sum dominance ratio and single dominance ratio as basis for determining dominant or important species. So by knowing the sum of this particular formula, then definitely makita nimo kung kinsay dominant dara na species. So aside from determining dominant, when you say dominant, kisa ang pinakadaghan dara nga species. So kung ang dominance niya, the dominant species are those that have the bigger DR values. So kung maibalaan mo na siya, maybe ang mga dagko nga dominant values are also important species in the area, which means kung less ng ilang population, then definitely it could affect the overall vegetation. So that's how vegetation analysis works. So Another is the quantitative vegetation parameters. So, kani, no, number of individuals or density is determined. So, abundance na siya. So, muna siyang pasabot na kung unsay mga naa nga present dara nga species, abundance, usa kadaghan. Then frequency, the number of times a species is recorded in a given number of small quadrats or at a given number of sampling points. So, ikapila siya ni balik sa isa ka quadrat. And then cover, either crown or shoot area or basal area. So, cover can be in the form of natay ginatawag na katong area kung labong ba kaayo siya, muna ang cover niya, in a term, other term is labong ba kayo siya, baga ba kayo ang iyahang cover nga murag sa isa ka quadrat tabon gid kaayo no magkugon grass gid ang nag-dominate, so kana siya cover niya, halos na cover ba niya tanan quadrat, so kana. Another is other measurable quantities such as height, stem diameter, and biomass. Leaf area index or leaf size and the bark thickness can also be used for quantitative vegetation parameter. So, kani sila na it involves a lot of computation and formula as well. So, let's now move on. Since we're dealing with vegetation analysis, it's not more on the ground species. We are also considering forest ecosystem. So, if we now mga vegetation magyapon na sila no kanang dili lang taga-deal sa mga sagbot-sagbot o katong mga naa sa ground na makita, but also the forest. There are also vegetation, no, plants gyapon na sila. So for sampling methods for forest ecosystem, we have two methods. We have the point-centered quarter method and the quadrat method. So I know that you are already familiar with quadrat method, pero ato lang yapon siyang i-review yapon, no? So for the centered quarter method, no, used to determine stem density through the measurement of tree distances. So the technique is developed based on the idea that the number of trees per unit area can be calculated from the average distance between trees. And it is very useful for estimating the volume of timber or timber inventory. So I have here, I drew a quadrat which will give you idea of what point-centered quarter method is. So ato na sang let's continue. All you have to do, or the materials needed or tools needed, is a compass, 30 to 50 meter rope, which is calibrated into 10 meters each, no? And then diameter tape, haga or abni hand level. So kana siya, pwede ninyo siya i-measure ang inyong kamot kung wala mo available nga tape measure dala nga kay you have to measure the DBH or diameter at breast height. So what to measure or record? You have the distance of trees from center, the DBH or diameter at breast height, the species, of course you have to record the species, kung sa mga species nga nakita dara, and of course the height. So procedure is establish a main transect line representing the whole study area, which may run a definite direction, maybe north to south, south to east, or east to west. So depende na sa inyo. You'll have katokong giingon nga katubitang mamili mong random lang tree, then mo din yung basihan nga muna siya ang inyuhang point. Maybe sa south, north, no? Kung asa mo mag-measure, kung unsay mga nearest nga mga plant species ang duol ana sa inyong gipili nga tree species. So along this main transect line, establish sub-transect lines perpendicular to the main transect line at a distance of about 20 meter or more. The minimum number of sub-transect lines divide the main transect into 4 quadrats. So, aside from that, no, we have here, within these sub-transect lines or sampling points, locate the nearest tree with DBH of 10 cm and above that falls in each quadrat. Then obtain or record the following. So 10 cm, no? Then distance, following distance of the tree from the sampling point. Identify the species. Then measure the diameter at breast height and measure the merchantable height. So, kaning point quarter technique is perhaps the most popular of the plotless sample techniques. No? Each sample is taken at a random location. So, for example, I'll show you a video of me. So, I made a drawing in which na A represents your chosen random point. This is along the transect line. So definitely area nga kaning you have to subdivide it into 4 imaginary quadrats. So north, south, east, west. So you have quadrat 1, quadrat 2, quadrat 3, and quadrat 4. So muna siya nga imaginary quadrats man na sila. So since you have to divide this in 4 imaginary quadrats, no, within each quadrat, unsa makita niyo ang nearest, what is the nearest tree will be included in the field sample. So there are 4 quadrats. You'll have to measure a total of 4 trees in each sample. So in quadrat A, as you can see, B is the first let's say inanayabotang nako dari quadrat 1, 2, 3, and 4. So here in my drawing, as you can see, this is your plot. Tapos sa inyong quadrat 1, you can see that kani ang inyong basis, inyong random nga plot is definitely ang pinakaduol is ang letter D compared with letter C. Kana. So definitely mao ni siya ang included nga letter sa imong sample point. Another is when you move to second transect, you can see that there is only 3 species. Kaning mga letter mga 3 species ni siya nga duol ani nga imuhang sample plot. No? So since this is the nearest to your sample point, then definitely this will be included sa measurement. So you have to measure the distance between your plot, sample plot, to the distance of these 3 species. So another is, for the third quadrat, you can see nga isa ra gyapon siya ka 3 species na belong dari. So you have to measure the distance between your sample plot to these 3 species. Then the fourth sampling plot, no, the fourth quadrat, I mean, is having 3 species. So, ang imuhang i-measure lang ang distance is the nearest to the sampling plot. So, as you can see, letter E is the nearest to the sampling plot. So, definitely, you have to measure your sampling plot to letter E, 3 species. So, that's how you do, what do you call this, point-centered quarter method. Okay, so now we move on to the procedure of computing. So as you can see, the total all points of plant distances for all species. So obtain the mean distance among species. So the total point of plant distance divided by the total number of plants. So Total number of plants no is katong mga species. Pila ka buok ang plants nga naa sa imuhang line. Okay, di ba? Before you conduct your point center quarter method, nag-establish kag line intercept or line transect in which nga gikalap nimo siya, kalap nimo ang isa ka area. So pagkalap nimo adto na apil sa imong transect ang katong sample point nga imong gipili, random sample point, then you have to determine katong nga line pila ka buok species, number of plants ang naa didto. So determine the mean area per plant. So mean area divide by plant. Then that's for the mean distance. Now you'll have the mean distance. Then obtain total density of all species. Total density of all species is the unit area in hectares divide the mean area per plant. So mo na siya, no? This is an example. This is a table example nga point centered quarter method with 5 sample points in Mount Makiling every 5-meter interval. So you have the elevation of 320 meters. So grabe na siya kataas. So as you can see, you have 5 sampling point. You have 5 sampling point, no? And then each sampling point has 4 quarter number. So kaning 4 quarter number mo ni siya tong kwadrat na duol sa imuhang sampling point. Kato sa 3 bitaw nga akong gidraw bitaw previously. So 4 ka buok, kay each quadrat isa ra man ka buok ang imong pilion no nga duol sa imuhang sampling point. Then definitely ang distance niya between kuan kaning sampling point to the first quadrat is the 3.5. Sunod 2.6, 1.2 meter, then 0.8 meter. Then unsa na nga mga species ang nakita ana sa first sampling point nimo? So you'll also have to measure the DBH or the diameter at breast height in cm. So if you have a tape measure, then you already know how to measure the DBH because you already had that in your ecology. So dapat unsay mga considerations on how to measure diameter at breast height. So dili na to ipa-measure tong kuan kasi tawag aning katong mga naay mga factors ana no, the height sa isa ka tawo kay not all height no, not all breast height are the same. So definitely kung taas ka, of course imong breast height will not be the same as others breast height. So usually the average lang gid nga height ang atong gina-consider for the DBH nga medyo reliable siya. So another sampling point no will also have 4 quadrat in which nga lahi-lahi pod nga mga particular species ang makita, no? So definitely in each sampling point there will be species that will of course be present, will also be present nga nadira no, mubalik siya, possibly mubalik siya. So another is the DBH no, having siya sa kadagko no, diameter at breast height, so dako gid tayo ang kaning mga trees no, maybe tigulang na ni sila or maybe mo pa na ilang juvenile nga measurement. So another is this one until you're done with all the sampling points you had. So that's it. You'll have that yes, no, nakita ninyo it's still the same. So the mean distance will be measured in a way that all of the sum of the distances from the sampling point to all the quarter number to all quadrat nearest to the sampling point, or the three species nearest to the sampling point will be summed up. So ang distance niya, 39.1, will be divided into 20 because 20 kabuk ang species nga nakita, regardless of the frequency. Like kanang kibali, for example, dari, kita ninyo nga si magabuyo present sa something point 2, tapos sa something point 3 present tapos siya. So it's the frequency na nagbalik siya. But you will not consider that as another species, but instead, gi-add mo siya as overall species, that you have 20 species at all sampling points. So that is 39.1 divided by 20 is equal to 1.96 meter. So that's the distance. So the number of trees per 100 square meter or meter squared is the area divided by mean distance. So 100, let's say yes, 100 ang atuang sampling na area, 100 square meters, then divide by 1.96 meter, which we obtained from the mean distance. So we have 26.04 square meter. No? So, kana siya. Then, we also have, I mean, it's not square meter, but meter, 26.04 meter. So, the absolute dominance is the mean basal area per tree times number of trees in species. So, we'll have that no? Later on. Sana siya pasabot. So, let's continue to table number 2. So, this indicates the number of trees per species in 100 meter or 100 square meter from the data in table 1. So as you can see, nga namin siya, number of quarters 4 over 20. Asa siya na-obtain? Number of quarters is, of course, let's say balobo, for example. What do you call this? Balobo, if we can try to count the balobo, let's say 1, 2, 3. Balobo 3 na ba? Naka may balobo da? Yes, 1. Oh, asa to na ko ang 4? 1, 2, 3, and then 4. That's why dani siya number of quarters 4 over 20. Where did we got the 20? It's the overall species na nakita sa all sampling points. So, ikapila ni balik si balobo of all sampling points divide by overall species found in all sampling points. So that is 0.2. Another is tamayuan. So tamayuan is 3 over 20. So ato na siyang tanawan na tamayuan. 1, tamayuan, 2, and then 3. So that's why we got 3 over 20 equivalent to 0.15. So now let's move on to Mount Tipanag. So since the number of quarters ay kapila siya ni balik sa tanan sampling points, once lang daw. So let's try to know if it's once lang gid siya. Mount Tipanag, 1. Say, Mount Tipanag, there's no presence of Mount Tipanag here. So definitely 1 over 20. 20 is the overall species again na nakita in all sampling points regardless of ikapila siya gibalik. So kadani naman siya ni balan ikapila siya gibalik of all sampling points in all quarters. So kasalang man siya, so that's 0.05. So now we know the number of trees in 100 square meters. So this 26.04, if we can recall, 26.04 is the number of trees per 100 square meters. So how did we obtain that again? Area divided by mean distance, this one, is equivalent to the number of trees per 100 square meter. So again, 4 divided by 20 is 0.2. So 0.2 times the number of trees in 100 square meter is 5.21. So ang balobo is 5.21. So adding that into, makita ninyo nga 26.04. It's because mo ani siyang estimation nato nga na ay presence ani nga mga species in this area, no? Sa number of trees, I mean in 100 square meter, 26.04 square meter ana having this type of mga species, having the presence of this type of species. So that indicates the presence of species in that area. So now let's move on to mean basal area by species in table 1. So I hope you're following, no? Even though medyo taas-taas na ta, but I hope you still understand what we are talking about here. So table 3, as you can see, we have different species available, no? And then the diameter, of course. If you can still remember, na how do we define diameter, circumference, basal area? No, we can use this figure. Circumference is the inches in which nga yung overall yun nga usa siya ka laparon sa iyang circumference, no? So siya katambok. Then diameter at breast height is the inches, in inches, no, is equivalent to the circumference plus 3.14 or pi. See, circumference plus pi. And the basal area, as you can see, kana siya, basal area in square feet, no, is the result of diameter at breast height times 0.005454. So that is how we obtain basal area. So as you can see, kaning breast height, no, is kana asara taman ang breast. Dili ang breast, regardless of the size of the breast, guys, ang height sa breast nato na of course in a regular height of a person. So we will not include the height katong mga taas nga na sa mga 6-footer kay lahi na ilang breast height ana. Kanang usual or average type of kanang average person's height. So that's it, no? And the absolute dominance is equivalent to mean basal area divided by 3 times number of trees of species. So that's how you obtain absolute dominance, or usa kadaghan ang makita ani nga particular species in an area. So this is how you obtain dominance, no, in that particular area. So dominance and rank of species in table 3. So here you can see that ang kaning mga dominance nila is definitely from obtain nga kung unsa kadaghan ang kaning mga species in that area. So knowing that Magabuyo ranked the first, followed by Balobo. So meaning ang pinakadaghan dito nga sa area nga imong gisampulan is the Magabuyo species. Having no nga daghan kayo siya, having 2,545.8 in dominance, no? That's it. So how about the absolute frequency per species in table 1? So as you can see, ang frequency is also the number, aside atong number or presence of katubit ng atong gi-talk about, if we can still remember the number of quarters, same magapon siya sa frequency, kapila man siya nibalik sa overall sampling point. Ang frequency is still the same, 5, kani siya 20 man, ikapila siya nibalik, kani
ikapila siya ni balik sa tanan-tanan species. Mo ni siya nga 4/20 out of 20 species nga nakita regardless nga ikapila siya ni balik sa kada katong kwadrat. Pero ang frequency is divide by 5. Since 4 divide by 5, ikapila siya ni balik sa sampling point. So if you can recall the table, di ba ang balobo? Okay, remember, balobo taha, species, balobo species. So 4 ang nakita, ika-4 siya nakita. Di ba ang balik-balik, when you say frequency, it's the balik-balik na kapila siya nag-occur, no, in a sampling point. So if we can recall this table, this one, sampling point, we have 5 sampling point, 1, 2, 3, 4, and 5. So, ingon si Balobo daw ko no, is having 4 frequencies. So, let's try to confirm. Balobo in sampling point 1, check. Balobo in sampling point 2, check. Balobo in sampling point 3, check. Balobo, sampling point 4, no presence. But in sampling point 5, there is a presence of balobo here. That's why it's now equivalent to 4/5 times 100. Okay, if we can recall frequency, it's the number of occurrence divided by the sampling point times times 100. So frequency is 80%. So it is dominated, no? The area is dominated with balobo species. So tamayuan na punta, no? 3/5. If you can recall 3/5, so let's now go back to the table again. So from the sampling point 1, tamayuan. So tamayuan is present in sampling point 1. Sa totoong tamayuan is 3 frequencies daw. So in sampling point 2, we have no presence of tamayuan. In sampling point 3, no presence of tamayuan again. In sampling point 4, there is a presence of tamayuan. And in sampling point 5, there is the presence of tamayuan, equivalent to 3 presence, no? 3 occurrence in the 5 sampling points, which makes it 3/5 times 100 is 60%. So on and so forth for Katmon, no? If you can go back to the table. So having total of 400 all in all. So that's for the frequency of all species in that area. So now, another procedure now we have left is how we compute for other parameters. So katong ganina, katong DBH or diameter at breast height, circumference divide by 3.14, 15, blah, blah, blah. I'm not, I can't memorize the pi. Then the basal area is circumference divided by 13.14, 15, blah, blah, etc. And then you also have to compute the relative density. Same nagyapan niya siya with the vegetation, no, underground. I mean, underground vegetation. Same with forest for 3 species, no? Relative dominance, and then the relative frequency, then importance value. So the importance value is the sum of the relative density, the dominance, and the frequency. So combination na siya. Importance value is defined as kung unsa kadaghan ang naadara. So kung dominant kaayo siya in that area, then definitely it should be given importance in a way that maybe this species, no, has provided a lot of kining importance in that particular area. Naa siya ginahatag importance in that particular area. Maybe in the soil type, maybe on the species present in the area, like animal species or insects, no, or pollinators maybe, or maybe dominated niya siya nga kani siya sa ana nga area. It's because it gives or it was dispersed by the pollinators. So inana siya. Na siya importance value. So now, measure or quantification of community diversity. So the simplest measure of community structure is a count of number of species occurring within the community is referred to as species richness. Unsa ka rich ang species? No, unsa ka daghan? Richness is yes, unsa ka daghan ang mga species in that area. So after counting all individuals of each species, no, so yun na to species richness na atay zebra, na atay lion, na atay fish, na atay ano siyang richness, daghan kayo particular species in that area, living in that area. So after counting all individuals of each species in a number of samples and determining what percentage each species contributes to the total number of individuals of all species, this measure is termed relative abundance of each species. So kung for example, na presence of zebra, lion, fish, blah, 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 then you have to do the relative abundance. Pila kabuok ang makita nga zebra, pila kabuok ang makita nga lions, pila kabuok ang makita nga fish in that area. So mao na siya. Excuse me. So next is a community with greater species richness and a more equitable distribution of individuals among the species. This characteristic of the community is referred to as species evenness. This can be shown by plotting relative abundance and rank abundance. So, kaning evenness it is defined kung asa siya na-distribute. Kung kani ba nga area having, let's say, 1 hectare, even ba siya pagka-distribute? Like, let's say, this is the quadrat of the 1 hectare kaning nasa inyong screen. Maybe kanibang lion is dani ra makita. Then the other species kay na din tanandari. So we don't call that species evenness. So definitely dili siya naka-abundant. So evenness defines in a way that species can be seen here, can be seen here, can be seen here, here, here. So meaning evenly distributed. So that's what we call species evenness. So muna siya no, kung naaba siya dari na sa tawag na distribute ba siya evenly sa isa ka hectare. So wala siyang species evenness. So definitely kung walay presence of particular species in every areas, then definitely they have less low ang ilang species evenness. So that's it. So values for relative density, relative abundance, frequency, and importance value per species in table 1. So you'll be able to know, no, if you can, pwede ninyo siya practice on table, no, na by computing the relative density and the relative dominance of the species, since it was already indicated the particular values of this species, no. So i-compute niyo relative density, relative dominance, no, para makapractice mo. So okay. So even the relative frequency and of course the importance value, and then try to know or try to confirm if balobo gyud ang rank 1. I think it's really rank 1. No? So if you can recall you can practice this. Okay, maybe this will be given as a quiz, maybe. So kamo na bahala. Okay. So the most widely used index of diversity that considers both species richness and evenness is the Shannon Index or Shannon-Weiner Index. No. So here is the formula for the Shannon-Weiner Index. H is equal to the summation of all species times the proportion of individuals, and then you multiply it to the natural log. So you need to have your calculator when you use this formula. Okay, so P1 no, is the relative abundance of each species. So kung usa ka daghan ng iyang species, summation na siya, then imo din siya i-inverse log. Inverse log. So that's it. As modified by Fernando, this is the biodiversity scale. So if ang imong computation reaches 3.5 to 4, then definitely the relative abundance and the evenness is very high. So kung inani ang imuhang kuan equivalent daghan kaayo siya species nga available dara. No, even kuan 3 to 3.49 is also considered as kuan high. So may balaan na to na siya by sampling and by computing all the relative abundance, relative dominance, frequency, and importance value. And by that way, makabalota kung unsa ka diverse ang area by knowing the Shannon-Weiner index. Okay. So another method, okay, since we already mentioned the point-centered quarter method, we also have the quadrat method. So I think you already know this, you are familiar with this. So only ang procedure niya determine the quadrat size. Ideally, determine the species are curved in the harvest method in sampling grasslands. However, it has been developed by ecologists to cut the methodology short. So suggest that we look for the largest species instead and simply double the canopy spread of that species to determine the quadrat size. So depende po na sa inyong scope kung sa kadako ang inyong unsay makaya sa inyong time and even your financial capacity. So another procedure we have here is to determine the number of quadrats. See to it that about 20 to 30% of the area is included and all vegetation patches are sampled. So when you say vegetation patches, so kung for example, na ay grass dari tapos wala dari, tapos na ay grass dari, you need to sample those having patches. So dili kay mamili mo nga danhirat na magsample kay dagan kayo dari maigo sa isa ka kwadrat, dagan kaayo nga dili mo i-sample tong patches. So it will be biased, no? So another procedure is gather all data desired from each quadrat. So all you have to do is to record all of this information, the name of place, the altitude, the slope, quadrat size, land use. So you have to define every term. Like for example, the land use and the slope. So imong definition sa slope, basin lahi ang definition sa uban. So maybe that particular slope is high to you, pero dili pa dayon siya in ana ka high for that kind of species living in that area. So yun na. Then species composition and number. So you have to determine or ID the species. So unsa na siya, unsay scientific name, local name, and even the common name. So mostly kung local name na siya, lahi ang term. But when you do common names, when you call it by common names or describe it or name it by common names, of course this has been widely used. Even international they use common names in identifying species. So kana. Plus the height, you also have to record the height and the circumference of trunk at breast height and other data you want to include like coordinates in your GPS. So coordinates like north, south, east, west. So having the coordinates kung asa gid dapit no? Then natin mo may balaan kung unsay altitude sa kana siya nga area. So naamo na sa tong GPS, no? I think you already know how to manipulate our GPS. So kana siya. You have to apply that in your thesis if ever you want to deal with biological sampling. No, biological sampling. So another is you have to tabulate your data, then compute the following: density, relative dominance, basal area, ratio of basal area height, relative, yes, no, frequency, yes, and all, species diversity. Then make a profile diagram based on height, mean distance, and mean data area. So, kana siya, no? So, next video will be all about sampling animal population in terrestrial ecosystems, because I still have to rest my voice since I've already talked maybe 2 hours, I guess. So we'll have to divide videos so that it won't be hard on your part, since everyone is also having a bad internet connection in their phones. So I'll be dividing my video lecture for the benefit of everyone. So this ends vegetation analysis. No, and I'll see you on my next video lecture for sampling animal populations in terrestrial ecosystem. So thank you. Bye.